in the last video, we looked at what metabolism is, and we looked at the two parts of metabolism. Anabolism, or building up, where we take small components and build larger products. And then we looked at catabolism, which are the breakdown reactions. And in this lecture video, we're going to take a closer look at a specific form of catabolism, the catabolism of carbohydrates. So, first of all, remember that we need enzymes for any of these reactions to occur. So there's going to be a lot of enzymes, and I'm going to kind of gloss over those, not really call those by name, but just keep in mind that they're there and they are very important. Okay. And our goal here is to break down glucose and produce ATP, which is the cell's, energy. Okay. And so this is our first step is glycolysis. So glycolysis, if we look at that word, glycolysis, it really is the splitting of glucose. So we're going to break down glucose. And this is the most common pathway for the breakdown of glucose. It's going to produce a little bit of energy, so it's going to produce a little bit of ATP. It's going to have some of our electron carriers that are going to be reduced. They're going to pick up some electrons. And then it also provides our precursor molecules for moving into cellular respiration, moving into the other processes associated with that. So all living organisms are going to carry out some form of glycolysis. It may not look exactly like what we're going to see here. And this is referred to as an anoxygenic pathway because it does not require oxygen to proceed. So glycolysis does not require oxygen. We're going to see other processes in the cellular metabolism or cellular respiration pathway that do require oxygen, but this first step, glycolysis, does not. Glycolysis is going to take place in the cytoplasm of both prokaryotic and eukaryotic cells. And we're going to take this 6-carbon glucose, and we're going to end up with 2 molecules of a specific 3-carbon sugar called pyruvate. Okay, so we're going to take glucose, we're going to end up with pyruvate. So this is a very simplified model or representation of what glycolysis is. So we start with 1 glucose molecule, and we're going to get this intermediate, and then we're going to get another intermediate, and finally end up with pyruvate here. Okay, so if we look at this process, we start with our 6-carbon sugar glucose, and this is going to require some energy. This is called the energy investment phase. We're going to put in 2 ATP. We're going to strip off those phosphate groups to end up with ADP. So this is that dephosphorylation that I talked about in the last video. We get these intermediates, okay, this glyceraldehyde 3-phosphate, and then we get what's called our energy payoff phase, or we're going to get some energy here. So NAD plus is going to pick up an electron to become NADH, and that's going to happen for each of our molecules here. And then we're going to put in 2 ADP and get 2 ATP out. So we're recharging, okay, we're phosphorylating our ADP to get ATP. So we're going to end up with 2 3-carbon molecules called pyruvate. Okay, so we start with glucose up here, have to put in 2 ATP for the process to begin, and then we end up with 2 pyruvates, 2 NADHs, okay, 1 for each pyruvate, and 2 ATP, again 1 for each pyruvate. Now keep in mind, again, enzymes are necessary for each of these steps to occur. Okay, we're just not talking about those, but just keep in mind that these reactions would not proceed without those enzymes. Okay, so at the end of glycolysis, we end up with 2 NADH molecules, 4 ATP, and 2 pyruvate. But we only get a net gain of 2 ATP. We end up with 4, but we had to put in 2 to make the process begin. So our net gain is 2 ATP. So 2 NADHs, 2 pyruvates, and 2 ATP is our net gain of glycolysis. Now for some cells, for some organisms, glycolysis is the end. We end up with those pyruvate molecules, and then that's the end. They take their 2 ATP and they're done. But in other cells, pyruvate can be further oxidized. So we can strip more electrons out of that pyruvate to drive other reactions. And so if we're going to move into that pathway, we have to go through a transition reaction, or sometimes referred to as the bridge reaction. And in this transition reaction, electrons are going to be transferred to the NAD plus to make some more NADH. And we're going to use this carrier compound called coenzyme A. For eukaryotes, this process is going to occur in the mitochondria. Remember, the mitochondria is the organelle that we refer to as the powerhouse of the cell. This is where those main reactions involving cellular respiration take place. In prokaryotes, who don't have mitochondria, this reaction is going to occur in the cytoplasm. So we've got this coenzyme A here. This is the molecule. It's a fairly large molecule. And then if we add this acetyl group here to it, then this becomes acetyl-CoA. And you can see by adding this acetyl group, we're changing the structure of this molecule. This phosphate group is changing structure. These phosphate groups are changing structure. So we're really just kind of transforming this coenzyme A structure. Now, once this takes place, once this reaction takes place, and again, this is not really showing you the reaction. This is just showing you the difference in the coenzyme A without its acetyl group, and then once it becomes acetyl-CoA, once that acetyl group is added. Then we can enter into what we refer to as the Krebs cycle. Sometimes this is referred to as the citric acid cycle or the tricarboxylic acid cycle, because citric acid is going to be involved in this process. So the Krebs cycle transfers remaining electrons from this acetyl group that's produced during this transition reaction to more of those energy carrier molecules, therefore reducing them. Okay, so let's look at our Krebs cycle. Okay, so here is acetyl-CoA coming in. All right, now you'll notice we only have a 2 carbon molecule here. Remember, pyruvate is 3. So in these transition reactions, we're stripping off a carbon. We're getting rid of a carbon and adding that acetyl, okay, to this CoA, and that's what's going to enter into the citric acid cycle. Now, in the citric acid cycle, we're going to strip off that CoA. We're going to end up with 3 NADHs, so we're reducing 3 of our NAD pluses, adding 3 electrons here. Our FAD becomes FADH2, which is the equivalent of adding 2 electrons here. We end up with 2 carbon dioxide molecules, which are going to be a waste product, essentially. And then we end up with 1 ATP. Okay. And this is a very simplified version. There are a lot more intermediate processes, intermediate molecules that are found here, and of course a lot of enzymes that are making these reactions occur. But we run the citric acid cycle twice, because remember we have two pyruvates that we ended up with at the end of glycolysis. So we're going to run this reaction twice. So at the end of our Krebs cycle, with both of our pyruvate molecules, we end up with 6 NADHs, 2 FADH2s. We end up with 4 carbon dioxides and 2 ATP. Okay. This figure shows the full Krebs cycle, or the full citric acid cycle. So you can see there's a lot happening here. We're adding carbons, taking away carbons. We're ending up with all of these little intermediate structures here. And cells can use these to produce different things. They can use them to make amino acids. They can use them to make fatty acids, nucleotides. So these intermediate compounds that are produced during the Krebs cycle can be used for other things. They're not going to waste. Now, the last step in the whole process, if we're trying to extract the maximum amount of ATP that we can, we're going to take the products of the Krebs cycle, and all of those electron carriers that have been reduced, that are carrying those electrons for us, and we're going to enter into a separate process called oxidative phosphorylation. Okay, oxidative phosphorylation. What's going to happen is we're going to take all those electrons that we collected during glycolysis, the transition reaction, and the Krebs cycle, and we're going to put those through a series of chemical reactions and produce ATP. Now, in eukaryotes, this process occurs in the inner membrane of the mitochondria. In prokaryotes, it occurs on the cell membrane. There are two main pathways that we can take in the electron transport system, which is the process that gets us to the main ATP that we're producing. We can have aerobic respiration, which requires oxygen. Oxygen is going to hang out at the end of the process and accept those electrons. It has a lot of redox potential. And we can produce more ATP in an aerobic pathway when oxygen is present. Another alternative is anaerobic respiration, where we're not going to use oxygen. We're going to use nitrate or nitrite, some type of nitrogen containing compound as the final electron acceptor. So aerobic is preferable, but sometimes aerobic can't happen. They don't have the appropriate electron carrier at the end called cytochrome oxidase. They don't have catalase, which can break down hydrogen peroxide, which can be dangerous to the cells, or there's just not enough oxygen. Our cells carry out aerobic respiration, but sometimes, especially if we're doing some type of anaerobic exercise, our cells run out of oxygen, and they'll switch over to anaerobic respiration, which is a more ineffective pathway. It doesn't generate as much ATP, which again is our goal here, to generate ATP for the cell to perform its normal reactions. Now, I'm not going to go into really any details because the electron transport system is very complicated, and it's not important that you know the details. What's important is that you understand that all of these electrons are going to drive this process, and this is an example of bacterial, the bacterial electron transport chain. And the end goal is to get ATP. That's our sole purpose. And in aerobic respiration, we can end up with a maximum of 32 ATP.